we'll get started. My name is Jack Dekkers. I'm faculty member in animal science at Iowa State University and one of the co-PIs on the Agricultural Genome to Phenome initiative funded by USDA NIFA. Yeah, it's my pleasure to host the field day today and hosting Dr. Amit Dhingra, who is a professor of genomics and biotechnology in the Department of Horticulture at Washington State University. And he actually served as, or is currently still serving as the interim chair of that department, but he's in the process of moving to Texas A&M, where he'll be take on the position of chair of the Department of Horticultural Sciences. And he's in the process of moving. So he's in a hotel room in Seattle, and he'll fly off to Texas with his family tomorrow morning. He'll also be joined by a couple of colleagues back in Pullman in the greenhouse, and he'll introduce them. But he's going to talk to us about breeding challenges in fruiting trees, and it's a topic of perennial crops we haven't dealt with. So we look forward to what he has to share with us. As always, please put any questions you have in the chat, and I'll look at those. And if there's a couple of breaks that will be inserted, but then also if there's a question that is pertinent to what is being talked about at that point, and I'll interrupt the speaker. So put your questions in the chat. So Amit, turn it over to you.
All right. I hope first of all, good morning from Seattle. I'm being joined by my colleagues. You can see Dr. Rishikesh Gogar. He's a postdoctoral associate in the lab. And then Nathan Tarlin to his right is our plant biotech manager. And he's been kind of the mainstay of a lot of the work that I'm presenting today. So they'll be joining me with their both expertise in video handling as well as subject matter experts as well. I'm going to be speaking about breeding challenges in perennial crops, and I'm very thankful to the organizers, Pat and Jack and Nicole, for giving me this opportunity and enabling us to highlight this often sideline topic of perennial crops, as the challenges are quite unique and somewhat difficult. But we've been our program has existed at WSU for about 15 years. And my history has been, I started working with rice, and then I went to tobacco and Arabidopsis, and I've gone back. I used to really enjoy the fact that rice takes about 6 months or 8 months in their life cycle. And of course, Arabidopsis is rather quick in a matter of weeks. So perhaps I was not satisfied and glutton for punishment. So now we work in terms of a few years or decades, if you will. But we've utilized some tools in greenhouses, and also trying to integrate the use of phenotyping, automated phenotyping, and also, of course, genotyping to be able to proceed along. So today, what I'm going to share with the group today is discuss with you some of the specific two case studies, basically. One that deals with breeding a wild Vaccinium species. A Vaccinium is Vaccinium, you know it as blueberry. There are wild Vaccinium species across the United States, and they have some unique traits. We are going to be speaking about Vaccinium membranaceum, which is unique to the Northwest. It has legendary fruit flavor and quality, and people forage a lot of that. So how do we bring on some of those traits into commercial blueberry cultivars, make hybrids? And the second one we're going to talk about is pear rootstocks, or developing dwarfing pear rootstocks. So with that I just wanted to, before I jump into the case study, I just wanted to say that, share the program with you a little bit. My program's mission is to produce nutritious food in abundance. I am myself a product of, I'm a product of the first green revolution, if you will. And what we focused on back in the 60s was trying to get enough calories. That was the main thing that we wanted to see. but now the goal for the world is not only subsistence, not only calories, but also look at nutrition, because that's one of the big pieces. So my program focuses on two main aspects: plastid biology, which allows us to look at function of plastids in fruit species, how the plastids turn into chloroplast to chromoplast, and what are some of the healthful aspects of that. Also focus on some photosynthesis engineering pieces. And in the post-harvest, we are working a lot on ripening. We have a novel pathway that we've kind of identified and working on characterizing that as we speak. And so we're working on these two areas, but it's all focused on fruits essentially. And we use all the possible tools that are there in our sort of toolbox to address these questions. And in all of that, of course, the other big piece that appears is one of the tools is breeding. And we've been employing greenhouse-based breeding. So I don't do this work alone. This is my team, current team at the top, and I'm standing here on the shoulders of the giants who have already graduated and contributed to the program in the past. So this work, what I'm going to present is part of that, what has been happening in the program, but I just wanted to start with acknowledging the team first. So we all kind of understand that there are major challenges with food availability. You've heard this over and over again, how many, 9 to 10 billion people. We need to be able to prevent 40% of food loss. So as I mentioned earlier, my program is focused quite a bit on plastid biology. So we've been characterizing how plastids develop in fruit. This is just a summary slide. You don't need to read anything about this. We are trying to work on understanding the novel role of some plastid targeted genes that provide more nutrition or even climate resilience for production. And some of these genes that we are characterizing by using chloroplast transgenics in tobacco, essentially, because it's hard to transform some other plant species. And on the left bottom, you can see an inset image of a lot of the phenomics tools we are trying to utilize, because when we perturb chloroplast function, the best way to characterize those plants are by utilizing these, measuring photosynthetic performance, or measuring how the electron flow is happening, and measuring fluorescence and non-photochemical quenching. The idea is to be able to identify proteins that might enable plants to grow better, and thereby engineer plants in the future as well. So that's one aspect of it. And then we have a novel AOX mediated regulation of ripening that we've identified. What you see here is a summary slide again. I'm not going to go into details, but we are identifying these pathways in existing cultivars. Pears represent a very unique plant species, which the fruit doesn't ripen off the tree or on the tree either. It has to undergo some cold exposure. So this is perhaps an adaptation in nature where they accumulate cold. You probably are familiar with this whole concept through vernalization, flowering in wheat quite a bit, but we are finding out that we can activate some of this ripening methods through exogenous application of glycolic acid. And we are trying to characterize the biochemical pathway in this process. So these are the two main focal areas, but as we are trying to develop these approaches, you can kind of see that it's all feeding towards how do we get more nutrition in our fruit as well. So we have some additional projects in the program. I'm going to talk about the first two on the left, huckleberry and blueberry breeding and the dwarf pear rootstocks, but we work on different areas, application of biotechnology, and underlying all of these projects is Nathan Tarlin, my colleague here, we've been working together for the last 14 years, developing these projects. He's the plant biotech manager, and currently another member on the team right now, Dr. Rishikesh Gogar. He is focusing on a lot of these biotech projects, most of all DNA-free gene editing that he's trying to establish in multiple species. So with that background, I just wanted to jump into talking about the first sort of case study today. So if you like blueberries, if you're interested in blueberries, it's a very interesting history. In the early 1900s, It was blueberry was discovered on an estate by one Mrs. White. She found these blueberries growing wild, and that's where the journey starts. And she found some germplasm. I heard this story from one of my colleagues. Mrs. White would tell her workers on the farm that whosoever finds the best, largest blueberry, she will reward that person, and that person has to take her to that bush, wherever that was. So she collected a lot of germplasm like this and passed it on to USDA. So the rest is history. Now the whole world grows blueberry. On the West Coast and other parts of the country, there are other Vaccinium wild species. They grow under forest shade. They don't grow out in the wild. In particular, Vaccinium membranaceum, the mountain huckleberry, or the black huckleberry. It has many names, depending on who you speak to. It is a traditional food of the native populations as well, Native Americans as well, and it's mostly foraged. There's another unique part about this Vaccinium membranaceum. It comes out as the first species after forest fires and controlled burns. So it has a place in this ecological progression. About 10 years ago, Nathan mentioned to me that nobody can transplant these plants from their natural environments and grow them in greenhouses. And I think that Nathan knows that's the key word to get me involved in a project. So when he said nobody else can do it, it's like, let's do it. So of course I knew, because if Nathan's bringing the project, he's already thought about trying to make it work. So all credit to him for making it work. So what I'm going to be sharing is quite a lot of his efforts in this process. So in the last few years, what has happened is Nathan was able to not only develop the tissue culture propagation process. So we were able to buy seedlings because this is a Native American plant, and we didn't want to be respectful of the First Nation people. We actually bought the seedlings from a native plant nursery, first 3 seedlings that he purchased, and then utilized that as a source to start developing in vitro processes where he was able to develop a tissue culture in vitro propagation process for huckleberries, and thereby he transplanted them into the greenhouse, successfully got them going. And one of the big things at the time was, will these plants flower in a greenhouse or not? That was a big question that was something to be addressed. And as you can see in our process over here, not only were they able to flower, they were also able to bear fruit, essentially the last photograph over here, as you can see. So the entire process we could replicate inside the greenhouse. And that's where the trigger started. Oh, now we can make crosses with commercial blueberries, because one of the big things with huckleberries is they have about 3 to 5 times more anthocyanin content in their fruit. They have special aromas and flavors that are missing in a blueberry. And everybody's kind of seen a blueberry fruit here on the left. Bottom, the mesocarp is translucent. There is no anthocyanin. The term blueberry comes only from the epicarp. So the idea was that could we create hybrids between the two? But then there were many challenges. Huckleberries and blueberries don't always flower at the same time. They are not synchronized flower. Their flowering time is not synchronized. They grow in different habitats. So by utilizing the process of cycling them through both cold, because these are temperate crops, they need to go through a dormancy period before they can flower. And it's taken many years of moving these pots into a cold room that we have adjacent to the greenhouse, bringing them back to synchronize the flowering between multiple types of blueberry parents and huckleberry parent and lines that we had, we were able to enable crop. We brought some bees into the greenhouse and we were able to get the crosses done. So it's some of the traits that we are interested in is flowering habit. Blueberries bear flowers in bunches, whereas huckleberries, as you can see on the right, only flower as single, they have single flowers. Fruit size is somewhat smaller. And the other part is that these huckleberries only grow in shade. Well, one could say that with the indoor agriculture, there are opportunities to not worry about growing them agronomically in a field. But definitely that's a desirable trait to have, because we will not transition our entire agriculture indoors. And the other main challenge, the other main target for us is anthracynum with mesocarp. I want to bring that back to the mission of the program, where we want to be able to grow nutritious food in abundance. So I think this is a good place to request my colleagues Rishi and Nathan to show some of these plants in the greenhouse, so we can do a little video tour of the greenhouse facilities. And I'll request, I'll stop sharing for now so that, or maybe Nicole, we can do a spotlight, and then I can continue to share the slides. Please advise.
I just did the spotlight and they are still small, at least on my screen. So I would say, yeah, stop share. There we go.
Okay.
Thank you.
So Nathan, would you please share with us some of the parents that we are using from Blueberry?
Yes, sure. Okay, so we'll start with the blueberry parents. This one we're looking at is called, it's a half high. So it itself is a hybrid between a high bush and a low bush blueberry. It's one of the parents. This is a southern hybrid type blueberry, called a rabbiteye type. It actually has 4 different wild species of vaccinium in its background. This one here, this is a typical standard high bush blueberry. We have 2 different types of high bush blueberries as parents, are actually 3 different types. This is one of them. It doesn't have any fruit on it right now. Over here is another one of the high bush type blueberries with a little bit of late fruit on it. This is also, it's a commercial high bush blueberry. It's a very typical type. Let's see. Here's another. This is another. I'll pick it up here. This is another hybrid high bush low bush called another half high type.
Well, Nathan, maybe Nathan, I just want to add at this point is that I hope you realize that they don't all flower at the same time. These are selected for production in different regions of the United States. They flower at different times, but in a way we are able to synchronize the flowering of all of this, utilizing environmental controlled conditions and providing them the chilling hours in a cold room. And a greenhouse is our tool to enable these wide crosses. Nathan, do you have any of the original lines for huckleberry that we purchased, and you had those plants sitting on by the wall, I guess, over there? Those are
no, yes.
Okay.
Yeah, I'm going to show you the huckleberry parents. These are clones of the original plants that you're looking at here. These are the mother huckleberry plants. Again, it's
How old are these?
late.
Yeah.
Sorry.
How old are these now? About these clones?
Oh, these clones now are about 6-7 years old.
Okay, thank you. And I also wanted to encourage the participants, if you have any questions, since this is a field day, please ask away any questions you may have, or you can type them in the chat inbox.
Yeah, Amit, I have a question. So yeah, you mentioned that each variety has its own requirements, but you have one greenhouse. So how do you accommodate that?
Well, we bring out the plants from the cold room at different time points we've kind of worked, I guess Nathan's kind of worked that timeline out. So that's his genius here to really, to be able to observe what the flowering times. And the good part is that when you read somewhere that a particular plant species requires 200 hours of cold conditioning at, say, 6 degrees Celsius. That's not exactly 200. We can go 300 hours, but there's a range. So by adjusting that time frame, we can really bring them all out at the same time. Also understanding once they are out of the cold room, it will take them 2-3 weeks to bud out, and then the flowers will follow. So I think it's been a learning process in this system, and he's able to then get all of them to flower. Not just what you're seeing over here right now, or he's showing you the blueberry parents, but you see a lot of other smaller plants. We will discuss that in a second, but I just wanted to take you. Step one was all the blueberry parents. Step two, I just mentioned to you, was the huckleberry original parental clones that we purchased. And then what he's showing you right now is that we are able to constantly produce, or over the years, we've been able to produce blueberries in a greenhouse. And it's rather interesting because I've been speaking to some of the indoor farming groups who are interested in producing blueberries indoors so that they can be available throughout the year. So unbeknownst to us, we were already preparing for this indoor agriculture that's knocking at our doors now. So I wanted to share my screen one more time so that we can continue the story a little bit. Right now, what we've shown you is I'm just sharing my slides. And Nathan and Rishi, please stay online because we will be discussing the next part as well. So what we've been able to show so far is that we are able to grow huckleberries in the greenhouse. They're able to flower and fruit. If we had done this presentation about, 4 to 6 weeks ago, maybe even 8 weeks ago, we would be looking at a full greenhouse full of huckleberries as well. But anyways, the photographs have to suffice a little bit. But what we've been able to show you, we can grow huckleberries in the greenhouse, they can flower, we can grow blueberries, we can synchronize the time. So thinking about a facility or environmental conditions for breeding these novel crops, this is what a greenhouse is enabling. One of the challenges is how do we determine if we can make crosses, and how do we screen those F1s in the process as well? How do we do rapid screening? We had to wait for the F1s to flower and fruit. It can take up to 3 years minimum. There is no quick process to overcome that issue. So that's something which I know that there are a lot of methodologies being utilized in other crops where people are using transcription factors like baby boom, et cetera, or utilizing the MADSBox genes, which we also utilize in apples, where a MADSBox gene is being utilized for constant flowering. So those are some transgenic intermediates that can be utilized to induce flowering to reduce generation time. But we are currently not using that in this particular case. So
maybe sorry, there's a
sorry.
question, Amit. So Pat Schnabel asked whether you can graft F1s to speed fruiting.
That's a great question. We haven't tested it yet. We will the blueberry parents also. If you start from a seed, it can take some time for them to flower. There is an inherent juvenility period. Rootstocks or precocious rootstocks are not something that we are aware of right now. Nathan, do you have anything else to add to that? Nope.
Sorry, please, can you repeat the question?
Yeah, so Nathan, I was asking question of Nathan is that, are there any precocious rootstocks that we know of in blueberries? I don't think so, but.
To my knowledge, blueberries are not grafted on rootstock. They're on their own roots. So I don't think that that exists.
But thanks to Pat's suggestion, Nathan, let's do a graft of these huckleberries on blueberries and see what happens. Now it's not me giving you a new project, it's Pat's idea. So.
Yeah, no, I was just reading the question. Can you graft F1s to speed flowering? That's a good question, and that's that's a possibility because the juvenility is about 5 or so years.
Yeah. Well, while the juvenility is 5 years, we've been able to actually, Nathan's been able to get them to flower within 3 because of this cold cycling, because we don't depend on outside cycles. And so that's been really helpful for us. But I just wanted to give you a closer look. The amount of, well, you can see the fruit size difference between a blueberry, a typical Chippewa one, as well as a huckleberry. Look at the huckleberry fruit. It's chock full of anthocyanins, and fruit size is distinctly small. However, the market price per, let's say, per pound, and I'll just say some random numbers. So if blueberries are selling for $5 a pound, huckleberries, which are usually forage, can go up to $50 to $70 a pound because of it's a very high value crop. And the other challenge is because foraging is a destructive process, or at least people who are not trained in it, a lot of the wild huckleberry stands are being destroyed. So there's a lot of challenge. So our goal is not just in trying to bring some of these healthful compounds and other benefits into a blueberry hybrid, huckleberry hybrid. We also, it will also have some ecological, hopefully ecological impact. We can't prevent people from foraging, but if the product is available in the market, our hope is that it will also prevent some of that ecological damage that is common. So here are some examples of the hybrids, and I think this is a great point. This is spring 2020 harvest. As you can see, we do have a large, in the middle, you will see a lot of fruit size and shapes, as one would expect. These are quite high bearing products. So this is unusual because we have these putative hybrids between blueberry and huckleberry. They're already flowering. We have done some molecular analysis to confirm that these are true hybrids. And now at this point, we are trying to sequence, utilizing sequencing-based approaches to confirm not only the hybrids, but as you saw, that it was kind of a common garden approach where we had all the parents of blueberries, blueberry parents and the huckleberry parents, and just let them cross. So we want to be able to also fix what are the parents, what are the male parents in these through genotyping processes. So some of these hybrids can be rather large fruited, so but they don't bear a lot of fruit. So this is obviously under we're cultivating them. So it's not that apparent what the yields would be, but there's been a lot of interest in this, and maybe the first approach would be to go into a greenhouse-based production in these. So at this point, I wanted to turn it back to Nathan and Rishi in the greenhouse. If you can show us some of the F1 hybrids with their variable leaf shapes, Nathan. I know you and I keep talking about, hey, this looks like that parent, and etc. But if you just take a global view, you will find that some plants leaves have developed anthocyanins, some haven't. So obviously these are segregating. The ones that are turning, the leaves are accumulating anthocyanin are turning towards dormancy as well sometimes. So we do know that maturity is another trait that is segregating in this, maturity timings. But I will let Nathan speak to you all about just different phenotypes that we're observing right now. Nathan, over to you.
Yeah, okay. So what I'm going to show you right here, this phenotype is very typical of wild type huckleberry. It is the wild type huckleberries at this point in the season are showing a little bit of anthocyanin as the leaves are getting, these plants are actually in autumn. So just like the wild type huckleberries, they look very much like the wild type huckleberries. If you'll notice here on a blueberry plant, blueberries don't typically turn that color. Well, some do, but at least in here, the ones we have, and in our seasons, they don't have that kind of coloring that the huckleberries have. So for example, here is one of our hybrids. Here was the first one I showed you that's looking very typical huckleberry-like. But here's one that is looking very different. You can see that there isn't any of the red coloring. The leaf shape is a little bit different. The leaf margins are a little bit different. The node spacing is closer. Well, actually, it's kind of in between the two. But this, I'm going to show you just sort of a selection of a range of phenotypes. This plant here is acting more compact, kind of, well, what we think is it's acting more like the, I showed you the sort of semi-dwarfs, the half-high plants. As we go, let's see if I can show it this way. As we go down the line, you can see we get even more compact, round kind of leaf shape.
And they are all the same age, by the way. Just want to make sure that we are all on the same page. These are all similar age plants started at the same time. So from
So
seed.
They are.
sorry, are they
Go ahead.
also F1s from the same cross, from the same parents, or different parents?
So the parents
Well,
is something we're determining with our genotyping process right now, but based on the, so for example, this particular one, I would say that this is the dwarf, you know, the short bush or low bush one, sorry.
Yeah.
I think
We know they
I think-
are all the same age. They're all the same seed lot, and they were all germinated at the same time, and they all come from the same mother plant.
we focused on the mother plant because we wanted to make sure that we are utilizing, because we want to bring the maternal germplasm forward. I wanted to point out, as you're looking at this in the context of the phenomes, genomes to phenomes, you can kind of see if there were an automated way to characterize these different pigments, automated, as well as leaf shape, leaf size. Of course, those phenomics information will be connected to the genomics. It allows us to then start associating some of those genotypes to the phenotypes, which can be really helpful in breeding. And it's a challenge. And also the other part is the shoestring budgets that we have to do these type of work. This is not a main crop. This is a minor crop, horticulture crop. So that is another challenge, which I don't think we can solve today or under this grant. Under this genomes to phenomes, but these are some challenges that that are faced by most minor crops. How do you really move fast forward? We have several phenotyping locations within WSU, but we're starting to do that. So what are you showing? You have a fruit there.
Yeah, this is one of the ones that's very late. That still has a few berries that are more huckleberry type, in the sense of that they're singular, but the huckleberries are long done by now. So this is also a variation in fruit ripening time. You can still see there's a few berries.
So there is a question, Nathan, I wanted to answer. Pat's asked a question. Do we already know whether anthocyanin accumulation in leaves is correlated with anthocyanin accumulation in the mesocarp of the fruit? So I think, Pat, all huckleberries, irrespective of the anthocyanin pigment accumulation in the leaves, will have anthocyanin in the mesocarp. These hybrids, oh, you meant the F1s? All the F1s so far, interestingly, have been like huckleberry. And this is where one of the hypotheses is that because huckleberry is a wild species, all the traits, and this, I don't have a basis to say that's the hypothesis we want to test, is all the traits of the huckleberry are the dominant traits. Because domestication, during domestication, we get rid of a lot of the dominant traits sometimes. And also, so that could be the reason. So that's our hypothesis right now, where we are seeing all F1s are almost like huckleberries, fruit, you know. There's another question, what is the ploidy of the huckleberry parent? It's been provided that different Vaccinium species are either diploids or tetraploids. In fact, Nathan and I were discussing this. There is no definitive answer to that question. And that's why we took this rather random approach of bringing some of the Vaccinium blueberry parents, which are both diploids and tetraploids, as well as the Vaccinum membranaceum. So this is almost like shoot first, ask questions later approach, where we created this common garden kind of I hope I'm utilizing that term correctly. It's a little bit of an adaptation of the common garden breeding approach, but we basically have all the parents in there of mixed ploidy as well as the F1s. So if I were to say that, if I were to, based on our understanding, they're perhaps diploid.
I'm showing you right now one of the parents that is the only one we have, well, one of the only ones we have that's a different species of huckleberry. This is actually Vaccinium ovatum. It is a wild species here in Washington, and it was commercially propagated about 100 years ago. Now it's mostly commercially propagated for landscaping. It's called the evergreen huckleberry, or did I say that? And it has very, very, very small little berries. But it's one of the most nutraceutical of the huckleberries. So that's why we included it as potential parent.
So our approach is that with all the, so the next question is, are you seeing improvement in stress tolerance? You know, these are, or stress tolerance, for example, drought, cold, disease, pests, and F1s. I guess the answer to that is those are to be determined. We are really at the initial stages of selecting some of these F1s and characterizing them in the second or third year of doing that. As we make some selections, we would utilize the, we will start as we move towards commercialization, we would start doing some of these tests for stress, et cetera. One of the things that we can do through phenomics is we have a couple of phenomics, enclosed chamber phenomics facilities to be able to simulate some of these conditions to at least screen. But as we all know, that what is done indoors doesn't always translate outside, primarily because of the light intensity. So we would be able to do these tests. So far, we don't see many pest issues because we have pest control, of course, in the greenhouse, pest and pathogen control in the greenhouse. But Nathan, we don't see any powdery mildew or other issues with these varieties or other fungal issues.
No, not really. We don't, and I don't see any at this point any real differences between the wild type and the F1s in terms of disease. But like you said, this is a pretty closed, controlled environment, and we do have integrated pest management where we use predator insects in here. So it's maybe not the right environment to exactly evaluate that trait. But generally, these plants, even the huckleberries, remarkably don't get diseases like powdery mildew or such in the greenhouse. Sometimes they'll get aphids, but that's more of a situation in the greenhouse.
Yeah, thanks. There's another question, and Nathan, you might have an answer. Is there any interest or market for Vaccinium parvifolium?
Well, that's a good question. I am not sure if there is if there's interest in or marketability in parvifolium. Is one of the more desirable huckleberry species. It's not the predominant species in our region. Yeah.
Oh, thank you.
So I know less about it, what its commercial demand is.
Yeah. Well, these are some great questions, and it's giving us many ideas as well. And as you kind of caught us in the middle of this breeding project, and I wanted to just highlight some of the challenges with a crop which has a juvenility time period from 3 to 5 years. And just wanted to also show you that this type of breeding won't be possible outdoors. It's just not feasible. So it allows us to really utilize the controlled environment conditions of land-grant university. You can see the greenhouses are all covered over also. That's part of the deal. But it allows, we are able to utilize the resources that we have to create this novel, unique project. Or right now we are considering taking some of the F1 selections and seeing if we could do a limited commercial release so that we don't have to wait 10, 15 years to release a final variety. There's another question. Are you looking into ways to estimate phenotypes at earlier data point? The first part of the question is, I'm trying to see what ways those would be. We have to observe flowering as such. And once we have the genotyping data, we could go after some seedlings that I think Nathan's just germinated from 2020 selections. So we don't have existing genotype to phenotype trait associations to go after those challenges. We do know that F1s are definitely going to look like huckleberries. We want to be able to do back crosses, either with a blueberry parent or a huckleberry parent. And that is the part of the genotyping work that is ongoing that can actually help us determine to go in that direction. There's a second part to the question, which is, can you induce flowering and berries? There are various transgenic approaches or various reports of utilizing the Matchbox gene or FT genes. We have stayed away from any transgenic approach, not because I'm opposed to it or anything. It's just we have lacked resources to go in that direction. But that's definitely a possibility. The way we are inducing flowering and berries right now is by cycling through them through cold. So we are able to get flowering by third, I should say, life cycle season, but actually it's because it's three calendar years, but in nature that might be 5 to 6 years overall. Another question is, could we do exogenous FT or something? My question back would be, does anybody have an experience by utilizing exogenous FT protein or inducing it transiently to induce flowering in any plant species? Does anybody have that experience? Because if there is, that would be a definite approach to utilize, and that would be something. There was a report from a colleague in Japan where they were able to do a viral infection in apple trees to induce flowering, or peach trees to induce flowering. We've been trying to get that vector in-house to see if we can utilize that. So no luck so far. But if there's any if there are any ideas, and the other part is that it might work in a model system or row crops. We should be able. We don't know if it will translate, but we are really open to trying some of those ideas. Next question is, has huckleberry been sequenced? The answer is not Membranaceum. However, blueberries have been sequenced. Definitely, Arcturus, we do definitely want to identify QTLs for breeding in the future. But definitely that's our goal. Right now, we are starting to sequence do a low pass sequencing, trying to utilize sort of a genotyping by sequencing approach to start finding these out and combining different platforms also. So we have both long reads and short reads, so that we can also start comparing it with the reference genome. And as all of us over here know, the age of reference genome has set. We need to be looking at individual sort of lines sometimes, because of each meiotic event creates a unique genotype, and also it creates different genomic sizes. So we're definitely interested in moving in that direction. In a way, if there are colleagues who are interested in this, or if there is an overall interest, we would love to collaborate as well. And Pat's question is, how is this research funded? Through donations, small donations right now. And we would like to see if there are other resources available. If you have any ideas, Pat, I would really appreciate that, since you asked me the question. So with that, I just wanted to say that this is case study 1, and we can continue our discussions, while I will request Nathan and Rishi to please walk over to our other case study, which is working with trees. So we're at 9:15, which is good time. We will have 45 minutes or 40 minutes for the next one, where we are going to discuss another major issue with perennial trees. Is that okay, Jack?
Yeah,
Nicole?
yeah, that's perfect. And while you do that, while you transition, question that I have, of course, you have to do this in the greenhouse because you have to make those distant crosses. But is the ultimate goal to produce a variety that can grow well in the field?
Yes, so we want to target that definitely. One of the challenges with huckleberry plants in general is that it will not grow. First of all, you can't even transplant it. But now that we've been able to develop, Nathan had to develop a method to get the right pH, right conditions in the pots, so those could be grown in the greenhouse. We want to have selections. So if you think about blueberries domesticated, we understand what its profile is, growth profile is. It's grown across the globe now, hot conditions, cold conditions. So we have that genetics, access to that domesticated genetics. What the genes are behind that, we don't understand. However, our goal is to identify F1 hybrids that we can cultivate just like blueberry, because it is both a high value crop for the farmer, but more importantly, it is a highly nutritious crop for human consumption. So yes, indeed, that is one way. However, that goal seems far away. So another aside is I've started a couple of companies as well. So I'm also thinking, if we are trying to do something, can we get it to market faster so there is an ROI for anybody who supports such a project in the industry? So we are speaking to some indoor farming groups where these hybrids could be grown indoors right now as well to start producing this crop. So that can also help in whenever we release something that can be grown properly, there will already be a market and traction in that case.
Yeah. And another question, is there a big difference in flavor and taste between the huckleberry, the blueberry, and the
Absolutely.
F1s?
Absolutely a big difference. Huckleberry flavor is, I think, for a lack of better term, and it doesn't really qualify properly, but I will call it legendary. Once you've tasted the huckleberry, it's utilized in ice cream. So if you drive in the Northwest during the early summer times, it is quite a bit of like a cult following, if you will. It's available for a short period of time. It's all foraged, and it's really a desirable fruit in this area.
Okay. Well, Pat just noted that Amazon has huckleberry jam for sale.
Yes.
So we can support Jeff Bezos's next flight into space, right?
And I have my colleague Holly Lane here, and Ferdinand's is our local dairy over here, WSU Dairy. Their huckleberry ice cream is available. If anybody ever visits WSU Pullman, you must try the huckleberry ice cream. I think it's available throughout the year, if I'm not mistaken.
Mm-hmm. And there's another question from Ismail about biostatistics. Are you using any kind of biostatistics analysis to predict those traits who may appear within the F1s? Because the reliance on the real experiments only, it may cost a long time period, as you said, at least 3 years. And if so, what are those main morphometric and morphological traits that your study is based on?
Great question, Ismail. Not at this point. As we generate our genomics data, as we start associating some of those traits. As you can see, the F1s don't segregate for much of the traits with the fruiting part that we're looking at. That's our main target right now. So our expectation is we have attempted some back crosses. As we get those progeny, that's where we're expecting some of the traits to come through. But yes, I just want to reiterate, this project might be 10 years old, but the challenge, one of the biggest challenge was first to make sure that we can grow these wild huckleberries in an artificial environment. That was the big piece. In fact, a lot of farmers from the area have come and seen, they want these large potted plants, and we've given away some of these potted plants as well. And it's kind of a novelty in that sense, but now, first of all, thanks to you guys for featuring this, because hopefully somebody in a funding agency hears about this and say, I'm willing to give you a few $1 million to take it forwards. So we can do all of those above things. In terms of your question about the traits, main morphometric and morphological traits, right now, the traits, as I kind of mentioned in my slide deck earlier, we are looking primarily if we can produce flowers in bunches. And also there's another challenge with perennial crops. Huckleberries will produce fruit on second-year wood, not on the first, that same season's growth, because they go dormant and produce shoots. Blueberries produce flowers and fruit on the new growth. So that is an important distinction as well. So you can kind of see we are trying to bring something completely out of the wild. But the good news is we have a vaccinium species that has been advanced quite a bit. So we are going through this process, and thereby by sequencing some of this population, we have a good population, which will segregate for some traits, at least F1s, to go in that direction. And Brenda just shared information that this ice cream won the taste competition at ASAS. I am not sure what that is. ASAS.
It's the American Society of Animal Science, which was held in hybrid form at Louisville, Kentucky last week. And
cool.
I was there and I tasted it, and it was very good.
So Jack, do you agree the term legendary describes it well?
Yeah.
All right. I'm going to share my screen again and kind of introduce you to the, oh, there's another question. Are there known wild blueberries with the huckleberry phenotype? We don't know. I think most of the blueberries that have been selected, well, okay, let me answer this question in a different way. Huckleberries are called as wild blueberries as well. So it's, but there are not, so I don't know how to answer that question, Okay. Well, thank you so much. I will share my screen and introduce another topic to you guys. You thought this was difficult? Let's go to the next one. Did I say we are glutton for punishment? All right. So now I'm going to talk a little bit about paired genomics, genetics, and breeding. This is a collaboration with my colleague, Professor Kate Evans. She's the one who released the most famous apple these days, Cosmic Crisp. If you haven't eaten it in your stores yet, please visit your local store, and putting a plug in for my colleague here. And Dr. Jessica Waite, who has just started at USDA recently in Wenatchee. So you may or may not be aware, but a lot of the trees, when you think about a fruit tree, you might be thinking of this 3-4-dimensional tree, 4 because time comes into the picture, which is all spread around. But in nature these days, that's what is true in nature. But when you come to actual orchard production, the trees have gone 2-dimensional. The concept has gone towards fruiting walls so that you can mechanize, you can make it more efficient. But that has only been true for apples. About 75% apple orchards in the United States have become fruiting walls. About 50% cherry orchards have become like that as well, but only less than 10% pear orchards have gone there. And part of the reason is we don't have a rootstock that will dwarf the architecture of apple. So in the 80s and 90s, when the apple industry was struggling, we in the United States adopted dwarfing rootstocks. Now, this is a term you have to understand. It's not just self-dwarfing or self-compact plant. It is a result of grafting, where you take a scion, which is highly vigorous, you graft it to a different selection or genotype of malus. And when you combine the two, the plant becomes dwarf. You can reduce the tree height to a few feet, 6, 15, 20 feet. 20 is still high, but there are super dwarfing rootstocks in apples. So while this revolution started in the 80s for pears, the apple people who are also pear growers are going, hey, we can find one for pears as well. Globally speaking, there are dwarfing rootstocks available for pears, but they don't adapt very well to the Northwest. Another factoid to consider, that over 80% or 90% of apple, pear, and cherry production in the United States happens on the West Coast. And Washington and Oregon actually account for almost 97% of the pear production in the country. It's not a very big industry. It's about $300 million industry, US farm gate value. And it has stayed like that for the last 3-4 decades, primarily because the pear varieties that we have, they have some challenges in being able to be consumed. There are many issues with that. The consumer is not able to get good quality pears. And another factoid, which is something interesting. You can kind of tell I'm a pear geek, so pardon me. But the pear varieties that we grow in the US, these include Anjos. You may have heard them as winter pears, or Bartletts, which are summer pears. And there's a third minor variety, which is a Comies, which is available for a very short time period. But then there are some other as well. Some of these three varieties that I just named were released almost during the French Revolution. And I'm not joking. They are over 200-year-old varieties that were released to the world, but the United States continues to produce them. We have not been able to switch to new cultivars because we are dealing with rootstocks that are non-dwarfing, that are not adapted to our environments. As I mentioned, there are rootstocks available, but they don't adapt very well. They're just not, and they're not compatible with the varieties that we grow in the US. So you might say, why don't you just get new varieties? Well, that has been tried. However, the rootstocks are not available, and it's kind of a chicken and egg story. However, after a lot of discussions in the last 15 years, we have finally been able to develop a program now collaboratively to move towards developing pear rootstocks in pears. So what does it take to really develop a rootstock? Just wanted to talk about that. So on the left, we are using 4 complementary approaches. I'll briefly discuss that, but what does it take to really develop a dwarfing pear rootstock? So of course, you need to have a germplasm collection that has that dwarfing trait. One of the primary challenges here is you can't just look at a plant and say, oh, this plant is compact and dwarf. It will impart that trait to the scion. Now you might say, well, the apple genome is sequenced. Why can't we utilize the information from there? So in apple, there are 3 loci known, DWF 1 to 3, known that cause dwarfing when you graft a scion on top of a rootstock. However, the challenging part is that the pear genome has been compared. We have found the syntenous regions. The challenge is that those markers or those regions don't contribute to dwarfing in Pyrus. So in Pyrus, a true dwarfing loci remains elusive. So we've taken a complementary approach where I'll now switch to the right-hand side. I'll go back and forth between these 2 diagrams briefly. What we've taken is we've taken this complementary approach where we are looking at 4 different types of resources to focus on this germplasm collection piece. One, we have a diverse set of germplasm where we are looking at 65 accessions that were identified across the globe, which provide various traits, disease resistance and other aspects. We don't know if any of them are dwarfing, by the way. We have no idea, but we are looking at these resources. The second one is that we had made some crosses between Bartlett Anjou and Bartlett Comies in my lab. These were done because with our experience in apple, we found when you make such crosses, you do find some compact plants, and we found some compact plants. Then the final one is this aneuploid population. So we've basically taken the pollen, gamma irradiated the pollen, and then done the crosses. So we were able to obtain about 49 seedlings of the 800 seeds that were obtained. That 49 seedlings survived. And this is one of the unique sort of germplasm sets we have. And finally, on the 4th is, this is Dr. Kate Evans's program. She's just making a lot of crosses between various parents, and some of the germplasm that we are utilizing in dwarf is being used as a parental as well. So going back to the left over here, we have completed the genotyping of these 3 types: the seedling populations, the diverse germplasm, and the replicated seedlings. As you would expect in an aneuploid, there is no point genotyping using a SNP array because aneuploidy will throw it all off because we want to basically do long read sequencing in this case. But anyway, that work is in progress as well. Once you've finished the genotyping, of course, on the other side, you need to propagate everything. You need to graft them to observe if they impart that trait. There is no shortcut to that, unfortunately. You have to do the phenotyping. and then do develop marker trait association, develop the tools, and do this process. So we are at this initial stages. So this project has also been ongoing since 2012. We've been working on, we made these first replicated seedling crosses in 2012. The aneuploids were crossed in 2013. So this is the population we're working with. Now, how do you take the, Pat was asking earlier about funding. I mean, these are 10, 20, 30-year projects to develop some of these things. Of course, we can use gene editing, but we don't know what genes to target in this particular case. So sometimes you have to have this sort of, we are hoping that the aneuploids will throw us the answers.
There's a question, what's the difference between a compact plant and a dwarf plant?
Both are synonymous, compact and dwarf, but I think the plant that is compact and dwarf on its own, if you graft a scion on top, it doesn't necessarily mean that a compact plant will make the scion on top also compact. And that is the challenge here. How do you bring dwarfing? And I'm speaking to the row crop group to some extent. You all know we've been able to bring dwarfing or compactness into wheat, rice, corn, everything. That's the same trait is eluding us in this case. So in apples, in cherries, we were able to achieve that same dwarfing, not by selecting a genetics that was self-compact, but dwarfing it by compacting, by grafting it onto a different type of selection. There are certain traits that you can score for. For example, one of the traits that is really cool is if the branch angle is obtuse or closer to 90 degrees, those type of plants which lack that type of apical dominance could be not only compacted themselves, but can also transmit that trait to the scion when grafted. And we have some selections in aneuploids where that is happening. This is a good timing to request Nathan and Rishi to come back online to give us a view of the plant material over there. Rishi?
Yeah.
Sorry, there's another question, follow-up question from Pat here. Is it correct that some dwarfing rootstocks produce full-size trees if they're not grafted?
You're right. They are full size. They're actually normally vigorous plants, and they are. It's a property of them coming together as grafted plants, composite plants. Absolutely correct. Thank you. Thank you, Pat. There's a question: is there any genetic influence by the rootstock over the productivity or adaptive traits in scions? Absolutely, Narayanan. There are so many examples documented in literature of the various traits that are influenced by dwarfing across the different genotypes. I think if any one of you, I'm sure many of you love wine, I just wanted to share with you that the entire French wine industry, I'm not making a claim personally, this is what I've been told, so if anybody's offended, sorry about that, but the entire French wine industry was saved back by a wild species of virus that was obtained in the US, which was resistant to a pest called phylloxera. And when they grafted the classical varieties, French varieties onto top of that, that's how that entire industry was saved. So not only it changes the agronomic properties, it can also, in certain cases like this, of course, rootstocks are selected for disease resistance. In apples, Cornell has released a lot of, recently released a lot of dwarfing rootstocks, but they're also resistant to replant disorder, replant disease disorder. So the rootstocks not only provide a lot of advantage to the scions in terms of dwarfing, creating more flowering traits, more flower and more productivity, they also prevent disease. One classic example that I learned 15 years ago was same cherry variety Bing, for example, when grafted on a mazzard rootstock, produces 2 to 3 flowering buds, but when put onto a Giesler rootstock, they can go up to 7 to 9. So Nathan is showing you clones of these aneuploid selections. They're all the same age. However, if you do a little bit of close-up, Nathan, you can show them some branch angles, perhaps, as well as the difference between internodes. Over to you.
Yeah, so this is a good example of where we have more of the 90-degree branch angle that's desirable. You can see like the secondary branches that are coming out that are more at that 90-degree branch angle, as opposed to, let's see, see, there was one, well, like for example, this is more of an acute branch angle, which is less desirable. But you can see, like in this group, that's more of a trait. Let's see. Let's come over here. This shows a real phenotypic difference. So this one is making very, very much right angle branches that are very long. Let me take one of these out. Okay.
So these
This
plants,
is
sorry, go ahead, Nathan.
this one here is acting more compact. Is it dwarfing? Well, that would remain to be seen.
So I think one point I wanted to highlight was is that these are clones from those 49 initial seedlings, and it has taken us a few years because we are doing this in a greenhouse. Another point I wanted to make is if we were to grow these clones outside, some of them may not have even survived. So it allows us to do this protected agricultural breeding, if you will, or horticultural breeding, where we can baby these plants, identify the right kinds, and ensure that we have the clones. So now we're getting ready to graft some of the scions that are grown in the US only on these plants. There is a question, any conjecture on why there is a correlation between branch angle and ability to act as a scion that can produce more trees? I think your question, ability to act as a rootstock. What we understand is that the branch angle is an indication of the apical dominance to some extent as well. While there is a correlation, this is I guess passed on wisdom from my colleagues and farmers. So a lot of the in this pairs you will see that a lot of these most of the pairs will try to grow at a very acute angle. The branches would like to grow at an acute angle if there's an example available somewhere. But in this prone type branching, we have reduced the apical dominance, thereby the branch angle is changing. Although there are other genes that obviously regulate that. That's just the sort of overall understanding. And the idea is that Jackson, with that type of phenotype, and this is something also, Gennaro Fazio, my colleague at Cornell, that's how he screened a lot of his initial rootstocks, which have turned out to be promising. Look at that. This is really beautiful. But also these because these are crosses between cultivated current varieties, Bartlett, Angio, and Comeys, the possibility is we may be able to find a compact pair scion tree, which could just be a varietal in itself. But these will take a few more years before they can fruit and flower. The average juvenility time in pears is, any guesses? How many years? You can place your answer on the chat for interactive purposes. 5, 8, 10. It's about 12 years, but
you want to get to guess?
Moira got close to 10, yes. So yeah, it is a pretty challenging trait, and there are some review articles from Gennaro's program where developing a rootstock is a 30-year process. You heard it right, 30-year process, because part of the trial is also to see if they can multiply these rootstocks in stool beds, because you once you identify a rootstock, you need to be able to mass produce it. Although I'm not speaking about it today, we've kind of addressed that challenge. I've started a spin-off where we can utilize in vitro propagation to combine various aspects of indoor farming, indoor growth, as well as we can really mass produce some of these rootstocks after we we can mass produce these plants. Indeed, Pat, it does make dairy cow breeding much speedier compared to that.
So to what degree can this compactness or morphology of the tree be affected by pruning and training the tree?
So of course, these are clones that you're seeing. We initially phenotyped the initial seedlings that they were growing. You can do somewhat of that, but pairs, the germplasm we are working with is quite wild in its, I guess it's not been domesticated much. Pyrus communis, at least in the US, has not been. So you can do a little bit, but it's real growth habit. There's a saying that pears, apples are grown on these trellises where you put the limbs on wires or branches on wires. Sorry, I do some farmer speak in between. Branches and limbs are synonymous. But we have seen that pear tree branch growth is so strong that it can actually lift or bend the wires very strongly. So the force is strong with this one. Sorry for that Star Wars thing, but we are dealing with a species that has remained untamed in terms of production. And that's why we thought that rather than just going through process, we should have this wild card mutation breeding in the mix as well. We don't know if these things will flower or fruit or not, but at least if we had a selection that can produce dwarfing, but also it's a great genetic resource for us. So we are currently, I'm going to share my screen one more time. We are currently in the process of, where we are trying to, sorry, where is that? Here. We are actually doing some genomic scans on this with long read sequencing to start identifying. So this is one of the workflows we have where we are able to look for the areas where we are seeing these larger differences. This is not from pears. This is from another colleague who's doing this in cherries. But I just wanted to show you the apple breeding timelines are about 18 years, just variety. Of course, aneuploids, this group understands aneuploids, but in pear rootstocks, that can be 30 years very easily. These were the initial phenotypes that we had observed for these aneuploids. The typical phenotypes, and these are juvenile. By the way, pear trees, when they're juvenile, can produce thorns. They are completely different once they become mature. So that is another challenge that we can't overcome with any type of quick changes. There's one more question. At what time point do you phenotype? Okay, I think I was already answering that. Is we phenotyped initially once we had the seedlings going just to see if there's any growth habits. And then all we want to, when the plants lose the thorns, then we know that they have not juvenile anymore. That's when we truly phenotype. So we've done branch angle measurements, caliper measurements, growth rate measurements at the same time. But all of that has been done in the greenhouse. I will add one more caveat. No matter what the size of the pots, these plants do become root bound. So they're not growing in their natural environments. So this is an approximation process. Will it translate or not to be seen? But in this process, we are developing methodologies to do this type of breeding in this process. It's a great question, Jackson. Thank you. I also wanted to show you some of these diverse accessions that we have from the clonal germplasm repository. You can kind of also, we're doing the same thing in this case, where we are multiplying, at least developing 20 clones per line, and thereby be able to then be able to come in and graft in a hope to identify that, are there any resources that provide dwarfing as a trait? And just these are some just examples, 6 examples of various leaf shape phenotype. This looks more like an olive tree sometimes, but you can kind of also see the different kind of leaf shapes plant. They are different calipers. They're really not ready to be grafted yet. I mean, one population might be ready like this, but the others are too small to be grafted. So there are a lot of challenges with this type of grafting, but I just wanted to highlight that. And a lot of this work was started with Marco Galli. He made all the crosses, and Daniel did a lot of the population structure analysis in these to demonstrate that these are truly aneuploids, as well as she did a lot of phenotyping, and Nathan's been kind of managing a lot of this in the greenhouse, and he makes plants alive and keeps them alive. But on the left, this pear tree is kind of an example of how different these looks like. I mean, most pears like to grow upwards. So we have some good selections. Now the litmus test would be to be able to go in and do the grafting to see if that happens. So of course, I have submitted these grants multiple times to AFRI, and of course everybody says, oh, it's very risky. Of course it is, but we got to do this because we are at a land grant university and we want to develop resources for our farmers here. So I also want to acknowledge research in my program is funded through many of these groups. I know we have time, but I just wanted to leave it for questions. And so thank you so much for this opportunity as well. But we can go back to the greenhouse and look for more plants if we want to, or open it up for additional questions.
Yeah, great. Thank you very much. Are there any questions? And you can put them in the chat, or you can just speak up and if you have any. So I'm not obviously not a plant breeder, but or you know, familiar with the grafting process. But is there any opportunity to graft between species? So to graft a pear on the apple dwarf rootstock?
Yes, that has been done. In fact, in pears, in our approach to really move fast forward, there's a Amelanchier species that's also rosaceae. Amelanchier is a service bush. It's a ornamental. So you can cross-graft Amelanchier with a Pyrus, and you can get flowering within 3 years. The challenge for commercial application, it's great for breeding, perhaps we can do that, and we're planning to do that. The challenge with that is that the caliper of an Amelanchier will remain very small. It's just a bush. However, the habit of a Pyrus is a tree. So we've seen some orchards. One of my colleagues, Todd Einhorn, who used to be at Michigan State and who used to be at Oregon State, is now at Michigan State. He actually developed an orchard. So the Pyrus sort of started engulfing the Amelanchier rootstock. I know that there are some selections that people are proposing to go forward with, but agronomically that becomes a challenge, production-wise, because then it will be a very fragile junction, and also it will create issues with future production, because the Pyrus can then throw off its own roots into the ground, so that the benefits of the rootstocks go away.
I see.
Yeah, so there's a question: what key phenotype traits and the opportunity of the greenhouse and field phenotyping? Johnny, could you elaborate your question? Maybe I'm not understanding your question.
Yeah, and go ahead, Johnny, unmute yourself and you can just speak.
Yeah, let's have a conversation. By the way, I appreciate all the questions. This is really a great discussion forum. Thank you. Oh, for apple phenotyping, I think right now Apple is quite advanced, but the main traits that we are looking, no worries, Johnny, audio is. So as far as I understand apple breeding, it's being done routinely. It's been done across the globe very easily. I think one of the challenges over there right now is focusing on the consumer, because if you think about it, we have over 5,000 known apple varieties. So the phenotyping right now is more routine. Will this plant have any disease issues? What's the branch angle? What's the architecture? Because you have different fruiting types. You can have spur type, or columnar type. So in that particular case, you can also look at, as we are trying to, depending on where you're selecting, if you go to the East Coast versus the West Coast, this is field-based phenotyping. I'm talking about the light intensity is more on the West Coast on average versus on the East Coast. So those are some challenges to look at, are these plants more productive? So I'm talking more about the photosynthetic performance type of phenotyping that could be utilized, that could be critical in that case. We also need to see one thing where I would say I have realized in the last few years, we often ignore what's underground, what's happening to the soil types. So that's, I think that is another piece that is being integrated now in our field-based phenotyping. What soil types are we at? What's our watering process and routine? So I think those are some things to be integrated in the process as well. I hope I've answered your question, Johnny, to some extent. We don't do breeding of apples in the greenhouse. Oh, okay. His question is how phenotyping will help the commercial production practice, such as pruning. I would say the answer to that would be, depending on what type of phenotyping. So I think we are trying to, in apples, we are trying to go towards automated pruning. But are you going to make decision-based pruning where a scout comes in, a scout robot comes in, looks at branch shapes and angles, and then comes? I think right now what they're trying to do is basically just hedge cutters, which are just walking, is just going around and renewing the wood. So I don't know if there is any, I personally not aware of that, but my colleagues at the Center of Precision Ag may be looking into that more. I have a general answer, but not a specific one for you. As I understand, they're just trying to bring these hedge cutters, which just go around the rows, and they're just trying to renew wood that way. Pat's question, my recollection is that quince can be used as a dwarfing rootstock for pear. Absolutely. However, quince is not cold tolerant. There are selections that are available for quince that may be cold tolerant, and those are being progressed. They do have compatibility issues. So you have to use an interstock, which increases the cost. Quince has been used as a parent. Also, there are some existing hybrids between quince and pyrus, but they don't, it's the adaptation piece. So there are a lot of resources globally also available that, advanced rootstocks from Italy, for example, and Europe. When we try to grow them in our environments, they just don't survive here. So another thing that we have to breed for the environment sometimes, especially where the industries, the other opportunity is to try some of these resources in other parts of the country so that we can expand the pear industry. Ismail has a question. Have you thought about the use of mycorrhization and its benefits? Not in this context. We are trying to utilize the application of biochar in some of the crops, which will foster different microbiomes. There's some research going on in my program in that case, but only, again, greenhouse-based research. That has been tried. There is a lot of organic farming that goes on in pears. However, it has not really changed the tree architecture that is the desirable outcome of this breeding process. It has provided anecdotal changes, but as you and I know, the next question comes in, what's the mechanism? How is this working? So some of those black boxes.
For my benefit, could you explain what mycorrhization is?
Well, mycorrhizae are basically fungal species, symbiotic or non-symbiotic, beneficial microbial fungal species that are present and that associate with the plant. And there are a lot of formulations available for organic production, not just for mycorrhizae, but also for microbes, beneficial microbes as well. So that's what mycorrhization in general. Ismail, I hope I described it okay.
I guess that's another very interesting angle to genomes to phenomes, right? It brings another, the microorganisms involved, just like in animals.
Yeah, Pat has a question. Will I be continuing this research in Texas? So Pat, I was actually excited because I've spent 15 years being a pear geek. I was excited to find out that there's an AgriLife extension station in Fredericksburg, west side, where it's a fruit production area. And the Pyrus is the species that can survive and thrive because the cotton root rot decimates apple trees. So in fact, some of these copies of these population I'm transferring between the universities. We're doing some arrangements to transfer a copy of my populations that we've developed over there, because that was one challenge in moving. What do I do with 15 years of research? So thankfully, both universities are great. They are allowing us to continue this work. In addition, there's coffee and pecans over there. So as I told you, there is 12 years of juvenility period in Pyrus, and they can grow up to 20, 30 feet. You all know that pecan trees can go up to 40 and 60 feet tall. So I told you I already like a punishment like that. So I might be interested in going after pecans.
This is just kind of a self-serving question. Are you still in Pullman? And if so, can I come see some of these projects in person before you go?
I am not in Pullman. However, we can coordinate with Rishi, and definitely you can take a trip to the greenhouse. My program will continue at WSU for 2 more years. I have to wrap up a few active grants, and in the meantime, slowly transition over to Texas. So Holly, you can reach out to anybody in the lab. They'll be happy to give you a tour. And anybody
Awesome.
else who's visiting WSU, if you want to take a look at these things. Great, thank you.
Are there other questions? We have a few more minutes. Amit, you mentioned that maybe is there still additional things to show in the greenhouse, or?
I don't know, Rishi, Nathan, are you guys done? Because I think we were just trying to show you some examples.
Yeah.
Essentially,
Yeah.
I think we are. Do we have, Nathan, do we have any of the diverse germplasm available here in this greenhouse?
They are dormant right now in the cold room.
Okay. Well, they're in a cold room, so we don't try to disturb them while they're there, and it's a little bit of a walk, so it won't be 4 minutes.
Yeah. Okay.
Yeah, there's not much to. They don't have their leaves right now. They're just dormant.
There's a question from Narayanan. How effective is mass marker selection in identifying suitable rootstocks? Are there any early selection markers available? Narayanan, as I was mentioning earlier, for apple dwarfing rootstock, there are 3 loci. There are markers available. Unfortunately, they don't translate to Pyrus. So in a way, we are trying to develop these germplasm resources right now to be able to develop some of those markers. And again, the challenge is to first, we are at a stage where we are now going to graft the scions onto these clones that we have, both from the aneuploids as well as from the diverse germplasm. And then the hope is that there will be some individuals that will provide that dwarfing trait or make the scion compact. And then we can really identify Pyrus-specific molecular markers that can allow us to do that breeding. Thank you. Thank you for your questions. Yeah.
Okay, I think maybe a good time to wrap things up. Nicole, if you want to share that final slide. And while we do that, I want to thank Amit very much for a very fascinating field day, and also Rishikesh and Nathan for their help with this. I think this is probably the closest we've been to a real field day, both in terms of visiting the greenhouse. We've done that before, but also the interaction. I think we had it was great to see the active interaction and the questions, and I think the only thing was missing is having the opportunity to taste the fruits, but we'll work. Zoom will have to work on that, but yeah, thank you very much for this presentation. And just for a couple of other activities, if you have additional ideas for field days, please let us know. Go to ag2pi.org. These field days every 3rd Wednesday of the month. We'll have another one in August. Tomorrow there will be another training workshop organized by AG2PI on phenomic data processing using machine learning, distributed computing, and container technology. So should be very interested. So sign up for that if you haven't yet. We were able to receive some additional funding from USDA NIFA, and all of that is going towards 2 more rounds of seed grants. And RFP for the second round was released last week, and the proposals will be due on the 19th. We will have a mini conference on August 12th to talk about how to apply for these seed grants and make some connections. As we're on that topic, I do want to thank, I see Ed Kalaiku is on. Thank NIFA very much for the support of these activities, and that are benefiting, the whole community. And I think this today was a good example of how we can all learn from each other. I learned a lot. Being in animal breeding, I learned a lot, and I'll never look at blueberries and huckleberries in the same way. And so with that, we'll close this. And actually, there's a couple of things in the chat. Let me see.
Just a few thank you notes, I guess.
Okay. Okay.
Very
Yeah.
grateful for the opportunity.
Yeah,
Thank you.
yeah. And the Slack channel is open. So if you want to continue the conversation, go to the Slack and yeah, notify your colleagues that may be interested, that weren't able to join, but that may be interested in looking at the recording. So thank you all very much, and talk to you next month.
Thank you.